What if you could cure diseases that have plagued humanity for millennia? Not by treating symptoms, but by editing the microscopic life living inside you. What if the solution to asthma, Alzheimer's, and even climate change wasn't in a pill, but in precisely editing the trillions of bacteria that call your body home? Jennifer Doudna didn't just help invent the technology that won her a Nobel Prize. In 2025, she achieved what seemed impossible just over a year ago, saving a dying baby with a treatment designed in 6 months. Now, she's pioneering an entirely new field funded with $70 million that could change everything we know about health, disease, and our planet. This is the story of CRISPR's next revolution. Let's quickly recap the revolution that started it all. CRISPR-Cas9 is a gene editing technology that works like molecular scissors with GPS. Scientists can program it to find any gene in the 3 billion letters of human DNA, cut it with precision, and then delete, repair, or replace it. Discovered by Jennifer Doudna and Emmanuel Charpentier in 2012, CRISPR transformed biology overnight. What used to take years and cost millions could suddenly be done in weeks for thousands. The impact has been staggering. In late 2023, the FDA approved Casgevy, the first CRISPR therapy for sickle cell disease. Patients who spent their lives in debilitating pain are now cured. As of early 2026, over 250 clinical trials are underway testing CRISPR for everything from cancer to heart disease. But here's what most people don't know. Editing human genes was just the beginning. Jennifer Doudna's path to changing the world started in Hilo, Hawaii, where a high school biology teacher handed her a book that would redirect her entire life, The Double Helix, by James Watson. She became fascinated by the idea that life's complexity emerged from something so elegantly simple, the structure of DNA. After earning her PhD from Harvard, Doudna spent decades studying RNA, the molecule that carries genetic instructions. Most scientists thought RNA research was a dead end. Doudna saw it differently. In 2012, working with French microbiologist Emmanuel Charpentier, Doudna published a paper that would change biology forever. They had figured out how to harness a bacterial immune system and turn it into a programmable gene editing tool. In 2020, Doudna and Charpentier won the Nobel Prize in chemistry. In August 2025, she was named the 2026 Priestley Medalist, the highest honor from the American Chemical Society. A new supercomputer at Lawrence Berkeley National Laboratory was named after her. But for Doudna, the real work was just beginning. In January 2026, she co-founded Aurora Therapeutics to make personalized CRISPR treatments available to millions with rare diseases. May 15, 2025. A medical team at Children's Hospital of Philadelphia held a press conference that made global headlines. They had just saved a baby's life using a treatment that didn't exist 6 months earlier. Baby KJ was born with CPS1 deficiency, a rare genetic disorder where toxic ammonia builds up in the blood, causing brain damage and death. The only cure was a liver transplant, but KJ was too young. Without treatment, he would die. But researchers at the Innovative Genomics Institute, Doudna's organization, along with teams from CHOP and Penn Medicine, did something unprecedented. They designed a personalized CRISPR therapy specifically for KJ's unique mutation. From diagnosis to FDA approval, 6 months. From concept to infusion, 8 months. In February 2025, when KJ was just 7 months old, he became the first person ever to receive a personalized CRISPR gene editing therapy designed for a single patient. The treatment worked. Within weeks, KJ could tolerate more protein in his diet. His medication doses were cut in half. He started hitting developmental milestones doctors thought he'd never reach. In June 2025, after 307 days in the hospital, KJ went home. Nature magazine named him one of the 10 people who shaped science in 2025. As an infant, this wasn't just a medical miracle. It was proof that personalized gene editing could move from concept to cure in months, not years. Here's something that might shock you. You are not entirely human. Your body contains roughly 38 trillion bacteria. That's more bacterial cells than human cells. They live in your gut, on your skin, in your mouth, and throughout your body. Collectively, this community of microorganisms is called microbiome, and it's not just along for the ride. Your microbiome helps digest food, produces vitamins, trains your immune system, affects your mood, and even influences your behavior. Scientists now know that an imbalanced microbiome, when harmful bacteria outnumber beneficial ones, contributes to an astonishing range of conditions: inflammatory bowel disease, obesity, depression, autism spectrum disorders, asthma, allergies, and even Alzheimer's disease. For decades, we've tried to fix microbiome problems with blunt instruments. Antibiotics kill bacteria indiscriminately, good and bad alike. Probiotics try to add good bacteria, but they often can't survive the journey or compete with established microbes. Fecal transplants work for some conditions but face acceptance and effectiveness challenges. What if, instead of killing everything or hoping something sticks, we could precisely edit the bacteria already living inside us, targeting only the harmful ones while leaving beneficial microbes untouched? Enter precision microbiome editing, Jennifer Doudna's newest frontier. In April 2023, Doudna and her longtime colleague Jill Banfield announced something extraordinary: a $70 million initiative funded by the Audacious Project, the largest scientific project they've ever funded. Their connection goes back to 2006, when Banfield first told Doudna about CRISPR systems in bacteria at a UC Berkeley cafe. Now they're combining their expertise: Doudna's CRISPR gene editing and Banfield's genome-resolved metagenomics, a technology that maps all the organisms in a microbiome and what they do. The goal? Precision microbiome editing to address global problems in climate and human health. Here's how it works. Step 1. Map the microbiome. Using genome-resolved metagenomics, scientists create a detailed blueprint of every microbial species present and their genetic functions. Less than 1% of microbial species can be cultured in labs, but this technology lets them study all of them. Step 2. Design the editor. Using CRISPR, researchers create genetic instructions that target only problematic bacteria, leaving helpful microbes untouched. AI helps design guide RNAs for maximum precision. Step 3. Deploy with bacteriophages. The CRISPR system is packaged into bacteriophages, viruses that naturally infect bacteria. Think of them as delivery trucks that only stop at specific addresses. Step 4. Edit or eliminate. Once inside target bacteria, CRISPR either disables harmful genes or kills the bacteria entirely, but only the ones you want gone. The result? A rebalanced microbiome that promotes health rather than disease. No collateral damage. No wiping out your entire bacterial ecosystem. This isn't theoretical. The research is happening now across UC Berkeley, UC Davis, and UC San Francisco, and the applications could transform medicine. Childhood asthma. 300 million children globally suffer from asthma. Research led by Sue Lynch at UCSF's Benioff Center for Microbiome Medicine has identified specific gut bacteria in infancy that trigger the immune dysfunction leading to asthma. The Audacious Project team is developing precision edits to remove these trigger bacteria before asthma develops. Unlike current treatments that only manage symptoms, this could prevent or cure the disease. Alzheimer's disease. Scientists have discovered that certain gut bacteria produce amyloid proteins, the same proteins that form plaques in Alzheimer's patients' brains. The gut-brain axis is real. and emerging research suggests that microbiome dysfunction may contribute to neurodegeneration years before symptoms appear. Precision editing could target amyloid-producing bacteria, potentially preventing Alzheimer's before it starts. Inflammatory bowel disease. Conditions like Crohn's disease and ulcerative colitis involve harmful bacteria triggering chronic inflammation. Instead of lifelong immune suppressants with severe side effects, imagine a one-time microbiome edit that removes the inflammatory triggers permanently. Obesity and metabolic disease. Your microbiome affects how efficiently you extract calories from food. Recent studies show certain bacteria make weight loss nearly impossible regardless of diet or exercise. Precision editing could rebalance metabolism at the microbial level, addressing the root cause rather than symptoms. The beauty of this approach? You're not adding foreign substances or permanently altering human DNA. You're restoring balance to an ecosystem that's already supposed to be there. But Doudna's vision extends beyond human health. She sees CRISPR as a tool to combat climate change as well. According to the UN Food and Agriculture Organization, 31% of all human-made greenhouse gas emissions come from agriculture, and microbes are at the center of it. Livestock methane reduction. Animal agriculture is the single largest source of methane emissions from human activity. But the methane doesn't come from cows directly. It comes from microbes in their guts. Current solutions like adding seaweed to feed work, but require daily interventions at enormous expense. It doesn't scale to the billions of grazing cattle worldwide. The audacious project's vision is an oral treatment given to a young calf that edits its gut microbiome once, providing a lifetime of low methane emissions. Research shows specific microbiome compositions can reduce methane by up to 80%. Carbon capture with enhanced soil microbes. Supported by the Chan Zuckerberg Initiative, IGI researchers are using CRISPR to enhance the natural ability of plants and soil microbes to capture carbon from the atmosphere. Agricultural soils have lost 487 gigatons of CO2 since modern agriculture began. The goal is to use farmland to capture and store carbon at a scale that meaningfully impacts climate change. Drought-resistant crops. IGI is developing rice varieties resistant to drought and disease using CRISPR, crucial for feeding a growing population amid climate chaos. They're currently in development. This is systems biology at planetary scale. We're not just treating individual patients, we're potentially treating Earth itself. Baby KJ's case proved personalized gene editing works, but it cost millions and required global collaboration. How could this possibly scale to the 350 million people worldwide living with rare diseases? In January 2026, just weeks ago, Doudna co-founded Aurora Therapeutics with Fyodor Urnov, who led KJ's treatment to solve exactly this problem. Aurora secured $16 million in seed funding from Menlo Ventures and is taking advantage of the FDA's new plausible mechanism pathway, a regulatory framework that lets them use data from one approval to rapidly gain clearances for related mutations. Here's the innovation. Aurora's technology uses AI to design guide-RN as that target different genetic mutations. They can keep over 99% of the drug the same and retarget it to individual mutations. Instead of developing one therapy over 10 years, they can develop dozens in parallel. Their first focus: inborn errors of metabolism like phenylketonuria, where patients can't break down critical nutrients. These affect millions, but have been considered too rare for traditional drug development. As Doudna said at Aurora's launch, CRISPR has always offered the promise of treating the root causes of genetic disease, but we lacked a scalable way to bring those therapies to patients with rare mutations. By innovating in both clinical development and approval pathways, Aurora is showing the true promise of gene editing for patients who were previously out of reach. The future of medicine isn't one size fits all. It's personalized, rapid, and scalable. The challenges ahead: ethics, ecology, and unintended consequences, ecological impact. What happens if we edit bacteria and they spread beyond their intended target? Microbes evolve rapidly and exchange genes. Could edited microbes create unforeseen ecological disruptions? The team is developing containment strategies, but nature is unpredictable. Equity and access. Baby KJ's treatment cost millions. Aurora aims to make it scalable, but will these therapies be available to everyone or only the wealthy? Could microbiome editing become another form of biological inequality? Regulatory challenges. How do we regulate something that's not quite a drug, not quite genetic therapy, and involves living organisms that reproduce and evolve? The FDA's plausible mechanism pathway is new and untested at scale. Long-term effects. Changing someone's microbiome could have multi-generational effects. What if altered bacteria pass to children through birth or spread to others? What are the unintended consequences we can't yet foresee? Food system disruption. If microbiome editing dramatically reduces agricultural emissions and increases yields, how will this affect global food economies? Who controls these technologies? Doudna has been vocal about the need for careful ethical consideration. The Innovative Genomics Institute isn't just advancing science, it's developing frameworks for responsible deployment. As she often says, the technology is advancing faster than our ability to fully understand its implications. That's both exciting and terrifying. We stand at a threshold moment in human history. Jennifer Doudna's work has given us the ability to precisely edit life at the molecular level. First, human genes. In 2025, personalized therapies designed in months. Now, the microbiomes that sustain us. Tomorrow, the microbial ecosystems that regulate our planet. This isn't science fiction. This is happening right now, in 2026. Aurora Therapeutics is developing therapies for rare diseases. The audacious project team is editing cow microbiomes to reduce methane. Clinical trials for asthma prevention through microbiome editing are in planning stages. Soil microbes engineered to capture carbon are being tested in agricultural fields. Within 5 years, we might see FDA-approved microbiome editing treatments. Within a decade, it could be as routine as taking antibiotics is today, but far more targeted and effective. The question isn't whether this technology will change the world, it's how we'll choose to use it. Will we cure diseases that have plagued humanity for millennia? Can we reverse climate damage we've created? Will we extend these benefits to everyone, or create new forms of inequality? The answers will shape not just our health, but the future of life on Earth. From a girl reading about DNA in Hawaii, to a Nobel laureate saving babies with personalized medicine, to pioneering a field that could heal both humans and the planet. Jennifer Doudna's journey shows us what's possible when curiosity meets courage. CRISPR was just the beginning. Baby KJ proved personalized gene editing works. Precision microbiome editing could be the revolution that finally unlocks solutions to problems we once thought insurmountable. The microscopic life inside you and all around you might just hold the key to everything. And it's happening right now in 2026. What do you think about editing the bacteria inside us to cure disease and combat climate change? Is precision microbiome editing the future we need? Share your thoughts in the comments below. If you want to stay updated on the cutting edge of science reshaping our world, subscribe and hit the notification bell. History is being made in real time, and you won't want to miss what comes next.